In today's video, we are going to see about chromosomes. If you find this video informative and useful, kindly like and subscribe my channel. Chromosomes are highly coiled ribbon-like structures formed by the condensation of chromatin fiber during cell division. So this is a cell. In this cell, you find the central nucleus here. Inside the nucleus, you find chromatin network or chromatin thread or chromatin fiber. These later condense to form chromosomes. So this happens during cell division. So during cell division only this process happens. So each chromosome contains a long DNA molecule associated with many proteins. So this complex of DNA and protein is called as chromatin. Chromatin substance, they are DNA and histones. So DNA is nothing but deoxyribonucleic acid and histones are proteins. A DNA molecule is very large and hence it is known as macromolecules. So the structure of DNA, it is composed of two strands wound around each other in a double helix. So this is one strand and the purple color is another strand. So both are wound together. It is like a twisted ladder. So each single DNA strand is composed of a repeating nucleotide which are made of three components: phosphate sugar arranged lengthwise, a nitrogen base attached to the sugar inward, and which is joined to the nitrogenous base from the other strand. So what it is composed of? So it is composed of, that is, there are four types of nitrogenous bases in the DNA. They are adenine, guanine, cytosine and thymine. So what happens is guanine will pair with cytosine and thymine with adenine. So it goes on. So this will be there here. So four such thing, again four such thing. So this will be going on. So they are given in detail here. This is a pentose sugar and it is a base here and it is linked with a hydrogen bond. So here with cytosine and guanine a triple bond, adenine and thymine a double bond. So these two strands together make a ladder-like arrangement with a nitrogenous base forming the rungs of the ladder. So it is like a twisted ladder. So what I said again, the same thing. There are four types of nitrogenous bases. So you should know that adenine, guanine, cytosine and thymine. So adenine always pairs with thymine. So it has a double bond between. Guanine always pairs with the cytosine. It's a triple bond. And next one is histones. These are proteins that help the in coiling and packaging of the DNA into structure units called nucleosomes. Now this DNA strand winds around a core of 8 histone proteins. Such a complex is called as nucleosome. So what a nucleosome means? When a DNA coils around, winds around the 8 histone protein, then the whole 8 histone protein you call as a nucleosome. Next one is structure of chromosome. So each chromosome consists of two sister chromatids joined along the length. At the point of attachment is called as centromere. So this is the structure of a chromosome. So this you called as a centromere. It is joined here. So these are the chromatids. So if this is a cell, the center is nucleus. Inside the nucleus, if you take, you find the chromatids in a network. Chromosomes you find in this way. So finally, if you take the structure of a chromosome, this is a chromatin fiber. It's a coil, highly coiled here. And these are the nucleosomes, the DNA, and this is the structure. So these two are the sister chromatids. This centromere plays an important role during cell division. It serves to attach the chromosome with the spindle fibers. So the equal distribution of chromosomes moves towards the opposite poles. This happens because of the centromere. One vertical half of the duplicated chromosome is called as a chromatid. So one such vertically, when it splits vertically, that one you called as a chromatid. Now DNA at a glance, we'll see what it is. So nucleic acids, there are two types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So components of the chromatin. So chromatin is made up of DNA and histones. And components of the nucleotide, pentose sugar, phosphate sugar and nitrogenous base. So what are the nitrogenous base? Purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are thymine, cytosine and uracil. nitrogenous base in DNA, adenine, guanine, thymine, and cytosine. Whereas in RNA, you don't find thymine, you find uracil instead. If you find this video informative and useful, kindly like and subscribe my channel.